Hello everyone, how are you all? कैसे हैं आप सभी लोग? I hope you all must be having a great time. And this is unit number 1 of paper number 2. We are going ahead. We have already completed 2 lectures. This is our lecture number 3, where we are going to study about the topics, that is chromosome structure, cell cycle and chromosomal aberrations. Now this way I have been showing you that this way it is written in your notification, but we have taken it out in a point format. In first lecture we have covered cell structure, function. I have told you that at the right time I'll be telling you about the cell cycle, and right now this lecture is the right time, guys. But before understanding cell structure, you have to go through, or basically in the second lecture we have understood about the genetic material, this particular point in detail, elaborative detail we have understood. Now over here we are going to understand, before cell cycle, the chromosome, its structure, and then cell cycle. After understanding this, you will be understanding that why these aberrations are happening in chromosome. And related to that, one more topic is there, that is polyploidy, euploidy and aneuploidy. ये इसी के साथ का टॉपिक है. This you will be finding under chromosomal aberrations only. But we will not be able to do it in this lecture because this is also a broad lecture which needs time. So we will be devoting certain time to polyploidy, euploidy and aneuploidy in the next lecture. After that we will be moving ahead with the laws of heredity, and after that we will be seeing. Okay? So without wasting any time, let us understand about the chromosomal structure, guys. तो सबसे पहले हम समझते हैं स्ट्रक्चर को क्रोमोसोम के. क्रोमोसोम के अभी तक बारे में मैंने आपको क्या बताया? व्हाट आई हैव टोल्ड यू टिल नाउ अबाउट क्रोमोसोम इज दैट दिस इज आवर सेल, दिस इज न्यूक्लियस, ओवर हियर न्यूक्लियोलस, एंड chromatin fiber is there. Chromatin fiber is not a chromosome. First of all, chromatin fiber is a structure like this, a fiber like form you will be finding. But at certain stage of cell division, when cell division takes place, and today we are going to understand this cell division, one phase comes, that is metaphase. During the metaphase, what you will be finding, that these chromatin fiber will be condensing like this, in this format, in this structure. And over here scientists were able to see the clear structure of chromosome, that how chromosome looks like. And this chromosome structure which we are understanding right now is this only, which we able to see metaphase condition under cell division, not naturally which is present in the chromatin fiber, right? This is under a certain stage you are getting, and that stage also I'll be showing you in an animated way. Don't worry about it. Okay? So our topic for today is chromosome structure. If you will be seeing the PYQs, see the PYQs. Only in the PYQ which have been provided over there, you will be finding that two things are being asked. One question will be directly on structure, second will be on operations. So don't worry about that. And rest, if one or two random questions have come, you don't have to worry about that, because we will be completing that in the enrichment. So don't worry about it, right? So let us see chromosome structure. When you will be understanding chromosome structure is somewhat like this you are going to find, guys. Like this you are going to find, right? Okay, this will be broad also over here. This will be broad like this it will be, and over here there is one part. Now different names will be coming over here. Try to understand carefully. So this particular part, outer layering which I have created, this is known as pellicle over here. Pellicle. Just understand the names first of all. Pellicle. This particular construction which I have made, this is a primary construction. And over here one important thing will be there, that is centromere. Like this particular type of bundle like structure you are going to see, and this we are going to call centromere. Ye aapko as it is, this you have to learn as it is. These are the terminologies for it. Now this is the structure, basic structure you are going to find. Primary construction is this. In the lower side you will be finding the secondary construction, right? Just understand the function right now. Secondary construction. Okay. Now over here, where is DNA? You will be finding like this. Over here you will be not exactly like this, but I'll be showing you. In this particular part you are going to find this DNA, and this is part which is where DNA is called up, is known as chromonema. Now the ends over here of this particular part of chromosome, these are known as telomere. Over here you are going to find telomere. ठीक है, telomere बोला जाता है इनको. And this particular full part over here will be known as chromatid. chromatid. ठीक है, chromatid is one arm, two arms you are seeing na with respect to center, one arm and the downward arm, it is known as chromatid. One arm is known as chromatid. So there is two chromatids present over here, right? This is one type of structure I am showing you, that chromosome is like this chromosome structure, and this structure is known like this over here: pellicle, chromonema, centromere, primary constriction, secondary constriction, telomere, chromatid. ये आपको पता होना चाहिए, right? And now I'll be telling you, showing you through the PPT also. See this structure over here, guys. ठीक है, क्रोमोसोम का अभी आप एक स्ट्रक्चर, वन आर्म आई हैव शोन यू बाय ओपनिंग अप लाइक दिस आर्म आई वाज शोइंग यू, राइट? This center you will be finding centromere written like this. Then you will be finding at the end telomeres. Both end will be known as telomere. This is a one chromosome. Chromosome are naturally present in duplicated form. Two forms you will be finding. I'll be telling you all about it. One arm you understand, and this arm is known as chromatid. So over here you will be finding chromatid also. That's why it is written as sister chromatids. One arm is long, one arm is short. ये हम बाद में देखेंगे. Later on we will be finding. One important thing you try to note over here, that this part over here, wherever centromere is this, suppose this is your chromosome structure which you have understood, like write this like this. Now I can draw. Now this is centromere over here, you have understood. Now near around centromere you will be finding one type of structure like this, and this structure is kinetochore. And the role of kinetochore I'll be telling you later on in cell cycle in today's class only. What happens over here, that I'll be talking about spindle fiber will emerge over here like this. Spindle fiber will be emerging and will be separating these two particular sister chromatids. Will be separating it. Spindle microtubules will be emerging over here. That I'll be showing you in the cell division also in today's lecture. Don't worry about it. But you try to remember that these type of structures are also there in the chromosome. Kinetochore. Okay. Now, if structure of chromosome directly they are asking you, if you will be seeing the P by Q. Over there you are going to find how you are going to write it over here, that the detailed structure becomes most visible during mitotic metaphase. Metaphase is written. What is mitotic metaphase? We will be understanding just after this. Because it is very important for you to understand first of all the structure of chromosome and what are these kinetochore, what is the centromere. Then only I will be able to tell you this cell cycle. Otherwise, when I will be telling you directly, I would have started with cell cycle, then it would have problematic for you to understand this chromosome structure. That is the sole reason I have left cell cycle for the end. Okay, pellicle, I have told you, thin outermost membrane covering of the chromosome over here. Encloses the matrix. What is matrix? Over here, this is chromosome. The inside material is a matrix over here. The same inside material of mitochondria is also known as matrix, right? Non-genetic appears during metaphase, you will be finding. Then there is matrix. Matrix, I have told you, it is a jelly-like substance enclosed by the pellicle. What it has provides internal support and holds the chromonemata. ठीक है, chromonemata, chromonema को ही बोलते हैं यहाँ पर हम. ठीक है, non-genetic. This is also non-genetic. This is genetic over here, chromonema, which I have told you like this over here, and the structure which you are seeing like this. ठीक है. So these are the thread-like coiled structure inside each chromatid. Inside each chromatid, inside each arm you are going to see. It was coined by Vejdovsky, if you can remember. That will be really good for you अगर remember कर पाओ तो in 1912. It is a gene bearing part of chromosome because over here only DNA is there, and it is visible during early prophase, you will be going to find. This structure may be coiled helically, forming primary and secondary coils over here, which we will be seeing a bit ahead. Then you are going to find chromatid. I have told you chromatid, one particular part, another chromatid like this. One of the two identical halves of the metaphase chromosome is known as chromatid. It is formed during the S phase of interphase by DNA replication. This also I will be telling you, same line I will be telling you in the cell cycle. So what you have to do when you will be understanding this cell cycle, again revisit this chromosome. In this way you will be having better understanding, guys. Okay? There are sister chromatids identical joint at the centromere. Like this you are going to find, I will be showing you also. Like this you will be finding one pair of chromosomes. Human have 33 chromosomes. Peas have 14 chromosomes. So 7 pairs over there. Right? 23 pairs of chromosome means like this over here. Okay? Non-sister chromatids belong to homologous chromosome over here. Right? What is this homologous chromosome? What is this heterologous chromosome? I'll be telling you. Right now, just understand the basic structure what I have told you. Centromere, this is also known as a primary constriction. Over here, constriction has happened. So this particular part is known as centromere. Very important constricted region where two sister chromatids are joined. essential for chromosome movement during cell division. It divides chromosome into two arms, contains two kinetochores. This shape protein structures you are going to find like this. I was telling you, spindle fibers attached to these kinetochores and stretch them out, and this will be dividing them like this. You will be finding. ठीक है, ये मैं सब आपको समझाऊंगा. This is done. Centromere is done. Pellicle is done. This you have to remember anyhow. Chromonema is done. Now telomere also you are going to find, which is stable terminal ends of a chromosome. It prevents fusion with other chromosomes due to polarity, protect chromosome integrity during replication. So this end which you are seeing, it remains like, it basically prevent fusion of other chromosomes. So it will remain, it will make it isolated form, right? Then there is secondary constriction, which I was telling you is it present in the end. And this is very important. This particular part which I was showing you like this. This is centromere and this is a secondary constriction. Over here, this is the site of nucleolus formation. Nucleolus will be formed. And that is why this is known as a nucleolus formation region or nucleolus organizer region. In the nucleus, you are finding this structure as nucleolus. Nucleolus is getting formed from this structure. So constriction other than centromere associated with the nucleus formation, nucleolus formation, not nucleus. Region beyond it is satellite region. Also it is known as satellite chromosome. SAT chromosome also it is known as, or also called as NOR. So this region which is a secondary constriction region is also known as NOR. Okay. So now you understand this information also, that chromosome was discovered by E. Strasburger, thread-like structure seen during the cell division. That too metaphase. बार बार सुनने को मिल रहा है. Metaphase बताऊंगा भी आपको. I'll be telling you that. The term coined by Waldeyer, discovered by Strasburger, term coined by Waldeyer. This you should remember by heart. ठीक है, by heart you have to remember this. Chromosomes have high consistency across generation, primary role in cell division and carrying genes over here. Now different species are having different chromosome. Maize is having 20, wheat 42, pea over here 14. Chromosome number they have identified it. Okay. And there is one more structure you are going to see is of chromomere. Chromomina I have told you. Chromomere is a bead-like structure visible only during the Pachytine stage of meiosis. ये ध्यान में रखो. Right, Pachytine stage of meiosis, when I'll be showing you, then again come to this particular part, just back my video, and over there you will be finding this particular thing, right? Again I'll be telling you in this lecture only. It is found in plants like maize, rice, salivary gland, chromosomes of insects. Not everywhere you will be finding these chromomeres. These are the regions of tightly coiled DNA and show characteristics bandings, right? Now you understand one more basic difference between homologous chromosome. देखो chromosomes how it is present in every species is, now you have understood that this is particular one part I have shown you, but chromosome is present in this form, right? In this form you will be finding one chromosome is present. Like this you are going to find one more chromosome identical to it will be present over here. I am not able to draw the identical picture, but you try to understand. Identical means their P length, their this chromatid lengths are same over here with respect to these two. So these two are sister chromatids, but these two are non-sister chromatids. इसको chromatids बोलते हैं ना हम. Right. So what you are understanding that these two chromosomes are particularly exactly similar to each other. So what you will be calling this homologous chromosome. And in the humans, you will be finding that somatic chromosomes are always homologous. Somatic means which are not giving us sexual characteristics, which are not responsible for sexual characteristics. They are primarily responsible for any other characteristic except sexual, right? The same things happened in other species too, but there are certain variation exceptions we will be understanding in this lecture in later on only in types of chromosome only. Okay? Right now you understand that this is one type of homologous chromosome. Another type you will be understanding where these two chromosomes will not be identical. Suppose not this particular chromosome is there, but this type of chromosome is there, a shorter one. So that means these are non-homologous. And non-homologous is also known as heterologous. That means these are not similar. And this usually happens in the gamete chromosomes. Gametic chromosomes over here. Right. sexual chromosome which are responsible for the sexual characteristics like the 23rd pair. Okay, this you will be finding. So see this over here, that in the somatic chromosome number we have 2n somatic cells, usually homologous chromosome. Whereas in gametic chromosome number we have only n, half of it. What is this half of it? How it comes? I'll be telling you. Exactly half of somatic number I'll be telling you later on, guys. अभी right now you have understood the structure. ठीक है, आप यहाँ पर हो, कहीं मत देखो, अभी यहाँ पर हो आप. This you have understood that I have a homologous chromosome like this, where everything is same, and the bars appearing like this over here, which you will be finding this like this, na? The bars which are appearing like this, these are the genes, way of representation of genes present on this particular part. Suppose this is A gene responsible for our hair color, then there is A gene should be there in this particular part. Okay, so these type of things you are finding with respect to this homologous chromosomes. Now what you have to find over here, that how the DNA is enclosed in this particular chromosome. This you have to understand, that how this chromosome organization has taken place. Okay, this chromosome organization, now I am telling you that how this chromosome is organized. Chromosome, how the chromosome are organized in this fashion, this we are trying to understand. And to explain this, we have one model, that is a nucleosome model. Nucleosome model. Again, let me tell you that this nucleosome is a new word which you are hearing right now. Nucleolus is something else, nucleus is something else, nucleotide is something else, a nucleosome you are going to see in this particular aspect. To form this chromosome which we have studied like this over here, to form this particular part, and later on in the cell it is present in the chromatin fiber type of structure, you have to understand this model. And this model explains you that you are this, in order to form this chromosome, there are three things which are needed over here. One, that is a DNA will be coiled up, so DNA will be needing. Second, over here you will be requiring a histone protein, which I was talking you in the first lecture and second lecture also. Now it is the time to understand histone protein also. And then there will be something non-histone proteins also will be containing over here. What will be the role? We will be understanding. Okay, these are the things which are needed. So what is going to happen over here is basically that this particular chromosome organization will be taking up in three processes. One is the conversion of DNA to nucleosome. Then the conversion of this nucleosome into solenoid. Then a conversion of this solenoid into chromosome. This is what we have to see. So these are happening in few stages. 1, 2, 3 stage you have our understanding. That if this is my DNA, now you try to understand. This is my DNA over here. Now DNA over here is like this. But now it is so huge, so long that it has to be condensed. Because this format में तो आप लगा ही नहीं पाओगे इसको. Right, so it has to be condensed. It has to be condensed. How? It will be condensed by changing its form, by changing its nature. So this particular pair, this particular DNA will be utilizing the histone protein over here. And histone protein are H1, H2, H3, H4. 4 types. But one more type you are going to find including over here, that is H2. Now you have 2 H2, so 2 H2 can't be there, we have to differentiate. So there is one H2A and H2B, this you have to remember. These are different proteins of this particular histone protein. Now they form one structure known as octamer over here. Octamer means 8 histone proteins will be aligning together to form the structure like this. Let me show you. Like this they will be forming one structure. These are happening at the nanoscale, right? So these are 4 histone protein, and alongside this you will be finding that more will be there. Like right, this 3D I am trying to draw. So over here you will be finding more histone protein, 4 more histone protein. So these are 4. These are 8 in total histone proteins. And what will be over here? 2 of H2A, 2 of H2B, 2 of H3 and 2 of H4. So 2 molecules will be there of these proteins. That's how this is becoming 8. That's why I am calling it octamer. This is the structure, protein structure of histone over here, which has been formed. Then where is H1 is utilized? H1 I'll be telling you. Right now what will be happening now, this DNA, now these things are as it is, scientists have understood this in this manner, right? So this DNA will be now coiling into this particular histone protein like this. So how many folds? 1.65 folds will be done over here. Folding से होगा क्या? Surface area reduce होगा, right? Condense होगा. So like this 1.65 fold will be done. Again, after that you will be finding from one area to another particular, again octamer, you will be finding one protein will be needed, and the protein will be needed over here will be H1, this particular, and this is known as a linker DNA. Linker DNA. I'll be showing you the images also, try to understand right now. So till now what you are understanding, that this has been formed, that means my DNA has been coiled up into this octamer which is present over here like this, and it is going, okay? Now there will be one more octamer which will be present like this. This is singular, I have told you. One more octamer will be present like this, okay? And my DNA will be coiled up like this, 1.65 fold linker DNA, 1.65 fold linker DNA, 1.65 fold. That means my DNA is getting condensed. Over here what you are finding, these are histone proteins. Now what you are finding like this, that this particular one unit of this particular thing, the coiled up DNA and 8 octamer of histone, is known as your nucleosome. This is nothing but nucleosome, right? Now these will be coming together over here. Nucleosome will be coming together. All these nucleosome will be coming together in large quantities. So you will be finding more will be packed over here like this. You will be finding like this. You are finding multiple folds are happening like this over here, like this over here. So what you are seeing? This is a condensed form, more condensed form, and this is a forming like solenoid. Solenoid, I hope you have seen in the 9th 10th class. Solenoid is a type of structure where coiling is done in this way. ठीक है, स्प्रिंग लाइक स्ट्रक्चर, मैग्नेटिक स्ट्रक्चर आप देखते हो, तो इसी फॉर्म में आपको डीएनए कॉइल्ड अप होता हुआ मिलेगा सोलेनॉइड की फॉर्म में. तो इसीलिए न्यूक्लियोसोम सेकंड स्टेज में सोलेनॉइड में कन्वर्ट होता है, और यहाँ पर आप देखोगे 30 टाइम्स फोल्ड हो जाएगा. देन सोलेनॉइड से क्रोमोसोम में जब कन्वर्ट होगा, यू विल बी फाइंडिंग 1000 टाइम्स फोल्ड हो जाएगा, राइट? लेट मी टेल यू ओवर हियर नाउ, लेट मी शो यू. देखो, सी दिस डायग्राम केयरफुली, दिस वन डायग्राम यू केयरफुली सी. दिस इज योर, कहाँ से देखना है? यहाँ से देखो, ये वाला एरो मैं बना रहा हूँ, यहाँ से देखना शुरू करो और कहीं नहीं देखना. ये डीएनए, ये डीएनए आपका डीएनए के अभी डायमीटर कितना है आपका? 2 नैनोमीटर है, ठीक है? 2 नैनोमीटर पे जा रहे हो आप. अभी आपको न्यूक्लियोसोम मिले. न्यूक्लियोसोम क्या है? ये ऑक्टामर है. ऑक्टामर किस चीज का? लेट मी शो यू. ये देखो, ऑक्टामेर किस चीज का है? ये है स्टोन प्रोटीन का ऑक्टामेर. अब यहाँ पर आप चेक करो, ये वाला डायग्राम चेक करो, अगर आपको दिख रहा हो. जस्ट वेट, है ना, लेट मी हाईलाइट ओवर हियर. आई होप यू कैन सी दिस. चेक इट नाउ. आई होप यू कैन सी दिस पर्टिकुलर पार्ट. अभी यहाँ पर क्या देखने को मिल रहा है? आपने ऑक्टामेर जो मैंने आपको बताया था. अब ये ऑक्टामेर 8 है. 8 में क्या? H2A, H2B, H3, H4. H1 नहीं होगा. इजी टू रिमेंबर. Now over here what you are going to find that there is a 1.65 fold which has been done of the DNA over here, which is 2 nanometer, but the fold has been done over here. Nucleosome is formed. Now from 2 nanometer its width has become now 10 nanometer. So likewise these folds will be happening over here. H1 will be the linker DNA, ठीक है, and they will be forming these nucleosome fiber. एक यूनिट को हम क्या बोलेंगे? न्यूक्लियोसोम. They will be forming like this, and this will be condensed into this solenoid form, like this solenoid you are seeing. And in solenoid what you will be finding, there again diameter will be increasing 30 nanometer. That means all this is containing this huge DNA. Then after that you will be finding this loop will be set up like this. Solenoid form में आपको loop मिलेगा. उसकी लेंथ कितनी हो जाएगी? 600 नैनोमीटर. एंड दिस वे ओनली, दिस इज नथिंग बट योर क्रोमैटिन फाइबर. इसमें में आपको दिखता है, और ये इसके अंदर नाउ इट विल बी कंडेंस्ड लाइक दिस, which the width over here you are finding 700 nanometer. Understanding? Let me show you another part where you are seeing this nucleosome. Nucleosome is formed, linker DNA is there, right? And then you are forming that this is solenoid getting formed, and a spring like structure is getting formed, 300 nanometer, and the condensation is happening like this over here. Now in between there is centromere which is connecting these two particular chromatids. Understanding? See this particular part also, you will be understanding in a better way. ये आया आपका डीएनए, ये आया आपका हिस्टोन प्रोटीन. डीएनए कॉइल अप हुआ और कंसिस्ट ऑफ 8 हिस्टोन मॉलिक्यूल्स. आई होप यू आर अंडरस्टैंडिंग. H1 से कनेक्ट किया, H1 से जो कनेक्ट किया, हाउ वी आर टेलिंग देम? लिंकर डीएनए. then you will be finding this, that the diameter is becoming 30 nanometer, then 300 nanometer, then 700 nanometer, then the chromosome you will be finding 1400 nanometer. So this you have to remember over here, from 2 nanometer, 30 nanometer in the stage of nucleosome, in the stage of solenoid 300 nanometer, and ultimately at the level of chromosome 1400 nanometer you will be going to find. This is the metaphase stage. Also metaphase stage I'll be telling you. So this is the nucleosome model of how chromosome is organized. I hope you must have got it. Now next topic which you have to understand over here is the types of chromosome. There are different types of chromosome, and these different types of chromosomes are on the basis of first number of, for the position of centromere. Second is the number of centromere present. On the position of centromere and the number of centromere which are present over here. Position of centromere means that what is the, where is the position of centromere inside the chromosome you will be find. Is centromere is present in equal way, that this particular P arm and Q arm is equal. There might be some structure like you will be finding where centromere is slightly offset. यहाँ पर terminology over here you have to use this terminology of offset, slightly offset like this. Or if you will be finding the chromosome is highly offset like this. And what do we call these type of chromosomes then? I'll be letting you know. ठीक है, अभी तो इस टाइप के स्ट्रक्चर्स आपको देखने को मिल सकते हैं, right? What in number of centromere, what you will be finding that how many centromere are present? Are only one centromere present or two centromere present? Are only one centromere present, two centromere present, or three centromere present? Like this it can happen. So let me show you first of all types of chromosomes on the basis of what centromere position. So position, if it as the center, then it is known as metacentric. If it is slightly offset, thoda sa offset is submetacentric. Highly offset, acrocentric. Then you will be finding at the end of this particular thing, so centromere is present, that is teleocentric. Thus no other arm is visible. Okay, this you have to remember: meta, submeta, acro, and teleo. So when you will be understanding the types of chromosome, one is on the basis of position of centromere, metacentric, submetacentric, acrocentric, teleocentric. Now few example you have to rectify if you can. Okay, for example, metacentric is seen in human chromosome 1 and 3. As it is, you remember it. Submetacentric you will be seeing in a human chromosome from 4 to 12. Acrocentric you see it in 13, 15. You remember only 2, 13, 15 or 21, 22. So I would recommend you to remember 21, 22 exactly in the example. Then teleocentric you will be finding this does not exist in humans at all. Alright, it is at the very end of the pole of the chromosome. On the basis of function also, if it is somatic, autosome; if it is non-somatic, sexual, then sex chromosome you will be finding easy. On the type of cells, on the basis of number of chromosomes. For example, if the chromosomes are containing half of the original cell, you will be finding these are haploid. That this also I'll be telling you that haploid chromosomes are also there. And if this is double, then diploid. If this is triple, then triploid you will be finding. Multiple chromosomes you will be finding, set of chromosomes, then this will be polyploidy. This you will be finding in a particular aspect, but not a very important division of cell. Okay? Based on a centromere number is very important. I have told you two division basically: centromere number and the position of centromere. Position of centromere you have seen: metacentric, submetacentric, teleocentric. Now over here, based on the number of centromere, if there is no centromere present, then we call it as acentric. If there is one centromere present, we call it monocentric. Double, dicentric. Triple, more than two, polycentric chromosome. More than two chromosome if it is present, then polycentric, right? So on the basis of centromere position, like this, you will be drawing the chart if they will be asking you type of centromere on the basis of position. Okay? If they will be asking you on the basis of number, then you have monocentric, dicentric, polycentric. This is the example. This is their definition over here. And how many chromosomes are present? Centromeres are present over here: 1, 2, or multiple. Okay? Now they are happening because of certain deformation like translocation, paracentric inversion, duplication, deletion, and translocation. Just remember it right now. I'll be telling you in aberration we will be starting. This exactly you have to learn over there. What is translocation, paracentric, deletion, duplication, translocation. This you have to study in a bit while completing cell cycle right now. Okay? Okay guys, so till now you have got the understanding of chromosome over here. What all understanding? I don't think now any part of the cell you are not understanding now. Okay, all has been completed, including the primary and secondary cell wall, including the plasma membrane over here, including over here the nucleus, endoplasmic reticulum, Golgi bodies, ribosomes, mitochondria, chloroplast. Under nucleus you have seen the nucleolus, you have seen the chromatin fiber. Chromatin fiber is containing nothing but these are chromosomes. In chromosomes you will be opening up, and you will be finding how you will be opening up now. You will be finding this solenoid structure, 300 nanometer. Then if you will open up more, now I can show you that these are coiled up in a octamer, and this is nothing but your nucleosomal model of chromosomes. And when you will be opening, you will be getting DNA. Like this DNA, you have understood DNA is made up of 3 parts: important phosphate, nucleus, that nitrogenous bases, and sugar group over here. This understanding you have made, guys. In this particular lecture, we have understood the chromosome structure. First of all, what is the chromosome structure? Pellets, centromere, chromonema, telomere. I hope you have understood. Then we have understood the types of chromosomes over here. How chromosomes is packaged over here, you have understood. Now it's the right time to understand the cell division. अब आपको cell के बारे में you know everything about the cell now. So it is wise for us to understand this cell division now. And a lot of details, a lot of things which you might be confused earlier with respect to cell division will be cleared in this particular aspect. After understanding cell division, we will be understanding why this aberration happens in DNA. And you will be finding that this topic we will be understanding under chromosome aberration. Right method or not? Chromosomal aberrations. Then we will be picking up this particular chromosomal aberration. This is the way I think it will be better for you to store the information. Otherwise, it will all be random, guys. ठीक है? जब हमने, when we have understood that what is cell, we will be understanding how cell divides, and in this cell division only these aberrations are taking place. What are all these aberrations? We will be understanding it. है ना? अभी cell cycle will be our focus. ठीक है? So now understand this cell division which I was talking about. Cell division, or you can also say it as cell cycle. Any species you take, any species, plant species, human species, right? We are concerned about these two right now. They have a system of replicating their cells. Now these two cells we can divide in 2 parts. Like one type of cells are needed to replicate the non-sexual parts. These are called the somatic parts, somatic cells. These are responsible for everything except the sexual cells. In the human category, you have gametes as sperms and eggs. In the plants category, you have gametes as, and this gametogenesis, sporogenesis, I'll be telling you. You will be having pollen and you will be having the egg in the case of female flower only, right? Female plant only you will be finding. Okay, that you are having the sexual cells over here. And these are also known as gametic cells, gametes. These are somatic cells, means non-sexual cells also you can say. In humans, height, color, eye color, hair, everything else which is there, needed regeneration, needed growth and development. So this is needed for growth and development. In plants, stems, roots, everywhere you will be finding these cells. Growth and development is happening. When growth and development is happening, the cell, the primary cell should be having the capability to multiply, and all the cells have the capability to multiply, guys, right? But when we are having these 2 types of cells, that means the multiplication should also be of 2 types. So the multiplication of somatic cells is known as by the process of mitosis is happened. Mitosis. And the multiplication of sexual cells happens by the process of meiosis. Very important. Now, why this particular aspect is having, like, why there are 2 types? I have told you that mitosis, what is going to happen? Mitosis. This is your somatic cell. This is your somatic cell again. from your mitosis, in mitosis, there is 2n chromosome present over here. There is 2n chromosome present over here. 2n is equal to 46 chromosomes in human, 14 in peas, 20 in maize. Like that, you are going to find the chromosome. This 2n represent diploid. We are a diploid organism, right? Whereas what you will be finding that from a parent, this is parent cell. In mitosis, we need of somatic cell that this particular parent cell will be converting exactly same. ये same नहीं बना, but you will be finding these two are identical and will be containing the same 2n number of chromosomes over here. Right, so we have to understand this mitosis. What is this mitosis and how it got replicated into 2? Whereas meiosis, we need the end product not to be as 2n, but n haploid we need. Because later on from mother and from father, this nn will fuse and become 2n. Right, that's why we need half the number. So from here, first 2 nuclei will be created or 2 cells will be created. That will be of n and n. But again, one more division will be happening in meiosis, where 4 daughter nuclei will be created of haploid. Understanding? So over here, 4 daughter nucleus or cells are being created from one somatic cell over here. और ये गैमीट ही है आपका, ठीक है, सोमैटिक सेल नहीं है, गैमीट है. लेकिन 2n से ये n की तरफ जा रहा है, whereas ये 2n से 2n ही आ रहा है, and over here you will be finding 2 daughter nuclei. Over here 4 daughter nuclei. ये बेसिक अभी डिफरेंस समझ में आ रहा है आपको माइटोसिस एंड मियोसिस में, राइट? Mitosis and meiosis में 1 or 2 difference you are understanding right now. More when we are going in detail now, mitosis, what is meiosis, then we will be understanding more about it, right? So mitosis you will be understanding is that step in the cell cycle. देखो cell cycle also, देखो this cell cycle what you will be understanding now. Cell cycle contains of basically 2 major steps. One major step is your interphase. This whole, if this is a cell cycle process, suppose this is circle is a cell cycle process. Cycle means cycle will be happening, complete from regeneration will be happening. So this particular till over here, starting from here until here, this whole phase is one part, and this is nothing but your interphase. And then after interphase, this process will be coming, and this process will be known as mitosis or meiosis. Okay, let me show you by the diagram. See this over here. Try to understand. Don't see anything else right now. Okay, suppose this is a circle. Circle starting from here. Circle starting from here. Okay, this is a whole circle you are observing. You are observing this is circle like this, like this, like this. Over here there is one mark, and then there is another mark. So over here, what you are finding, that this whole phase is known as interphase. This long 80%, 90% of the time you are in interphase. Abhi aur kahin mat dekhna, no, don't see anywhere else right now. Then you are finding there are 2 stages which is considered to be a mitotic phase only, in which mitosis and cytokinesis will be happening in these 2 stages. So this is a very small space where this mitosis is happening. Otherwise, the cell is in its interphase most of the times, 90% of the times, and then 10% of the times now it will be going through mitosis. The cell cycle, what you will be understanding is understanding the pre-mitotic events that is happening in a cell, right? So now this, what you are finding, this interphase over here, this interphase, and then mitosis will be happening. So interphase is divided between over here G1, G2, and in between this S, G1, S, G2. Okay, these are the phases under interphase. Now one by one we have to understand this particular part. When they are asking you about cell cycle, you will mention about this in the interphase. When they are asking you about mitosis, you will only mention about mitosis, right? So let us see this, what is happening over here. The cell cycle contains 4 phases: G1 phase, S phase, G2 phase. This is your interphase, and this M phase is mitotic phase. Understanding this must, either you can write as interphase and mitotic phase, or further divide by showing it like G1, S1, G1, S phase, and G2 phase, right? Now understand that what happens in these phases. G1 phase you will be finding that it is the first phase of the cycle, right? Over here, what is going to happen? This cycle will be starting, and in this, the cell size grows over here. So cell will be growing in size, right? It will be growing in size structurally and functionally, and matures into a parent cell, because now cell is going to get ready to multiply, है ना? So at some time during this interphase, the cell receives signals that it should go division again. These signals led to the cell to enter the next phase of the cell cycle, the S phase. देखो, G1 phase is the preparatory phase. Whole interphase is the preparatory phase. Under G1 phase, this will be becoming larger in size. All things will be becoming larger in size over here, right? So in this phase, you will be finding these structures will be getting bigger, and also these will be multiplying. The whole cells except the nucleus will be multiplying over here. Next, the most important phase, S phase, you will be finding it is the synthetic phase. The cell undergoes DNA replication. The most important thing over here is that in the nucleus, DNA will replicate over here. As a result, chromosomes in the cell consist of two identical chromatids. The nuclear material in the cell is doubled. Only the nuclear material will be doubled. For example, if the chromosome number is 23 or 46 over here, and in this particular chromosome, suppose there are 100 DNA molecule which are present. So this 100 will be converted into 200. Understanding, DNA will be getting doubled, not chromosomes will be getting doubled. ये थोड़ा सा confusion रहता है, just clear it out. So what you will be finding over here is this DNA will be getting doubled in the S phase. You have to remember this is also known as synthetic phase. ठीक है, तो over here DNA synthesis also written. Over here cell growth will be happening. Then cell growth, G2 phase will be coming, and G2 phase also you will be finding that in this also it will be preparing itself for cell duplication, and the duplicated DNA is checked for errors. Proteins necessary for cell divisions are made at an exponential rate. हर चीज double चाहिए, everything you need double now. So that will all again be checked over here. Then the cell receives signals to undergo cell division and enters the M phase. And M phase is nothing but your mitotic phase. It is a phase of the cell during which the nuclear division takes place. एक से दो nuclear division होने वाला है. So this is really very amazing. These things have already occurring in our system, right? But scientists have understood it and explained it in a better way. Okay, so this particular part you have understood what is happening in interface G1 S and G2. Then you are coming to this mitosis phase. Now you understand the mitosis. I'll be showing you the animation also over here. Remember, under mitosis there are certain phases which are going to come, and this is P, M, A, T. This P is prophase, this M is metaphase, this A is anaphase, and this T is telophase or telophase. Understanding? So one by one our cell will be going from this direction to this direction. Prophase, metaphase, anaphase, telophase. Okay, now you understand each phase over here, guys. First of all, let us talk about the prophase. You have interphase over here. Interphase, you have understood the G1, S and G2. Overall, you have understood that over here the cell parts are getting duplicated. In S phase, the DNA got duplicated. Suppose this is a chromatin fiber, it got duplicated. And red, I am showing you that duplicated part. Now you are going to the prophase. Now prophase also consists of certain phases. Under this, certain stages will be coming. This is early, this is middle, and this is late prophase. What is happening over here is primarily you will be finding that in this particular part, prophase mitotic division has started. This particular nucleus will be condensing, guys, over here. Suppose this is one DNA material. This is one DNA material. This will be uncoiling and getting smoothened out, will be getting condensed, and nucleolus will be also there, right? In middle phase, what is going to happen? In middle phase also they will be again over here will be condensing like this. Condensing over here like this. Just let me show. Okay, they will be getting separate over here. Like this, this is over here. This is another duplicated part. So there will be clear-cut separation you will be seeing between the two DNA parts. Duplicated one and the original one. ठीक है. In the late phase, what you are going to find is slowly and slowly this particular nuclear membrane and this nucleus will go away. They will disappear. So it will look like this over here, like this, and over here like this. Okay, I'll be showing you too, don't worry. Okay, so in prophase you have understood there is early phase, middle phase and later phase. Overall what is going to happen, the duplicated part will be going to see very clearly over here, very clearly. Nucleolus will be disappearing at the later stage and nuclear membrane also will be disappearing. So what will be there? Only these two duplicated chromosome and other organelles are also duplicated over here. In the metaphase, what you are going to find that in the metaphase, your centrioles will be coming into picture, centrosome in plants and centrioles in animal cell you will be finding. In metaphase, you are going to find these two particular parts like this. And from here, spindle fibers will be emerging. And spindle fibers are nothing but made up of this tubulin protein. Only remember the words, you have to put down the words, right? So from here, these tubulin protein will be coming like this. And these chromosomes over here will be arranging in this fashion, guys. Metaphase. Now remember chromosome, that when we study about chromosome, इसको थोड़ा सा और clearly मैं दिखाता हूँ. Suppose this is your cell over here. ठीक है. Now you will be finding that this is a metaphase. So your chromosome will be forming like this, like this, like this, like this, and there will be around 20 more, right? If we are taking any species. Now I have told you that there is a duplicated one too. So duplicated one will also arrange. Duplicated chromosome is also arranging. Over here there is centrosomes in plants. And from centrosomes you will be finding spindle fibers are coming. So like this spindle fiber will be attaching to the kinetochores. Kinetochore, now just remember the starting of it. Not like this. One part will be attached to only one kind of chromosome like this, like this, like this. And on the white you will be finding that this will be attached, this will be attached, this will be attached, this will be. And in the anaphase now you will be finding that these two will be separated over here. So how this will be looking like? That these two will be separated over here. Suppose over here I am showing you. Then the yellow part will be going like this over here. Will be taking the part over here. And the white part will be coming over here. White chromosome, the duplicated one. They will be assembling over here. And in the anaphase now suddenly you will be finding the centrosome will be coming, nucleolus will be emerging, and a nuclear membrane will also be emerging like this. In the teleophase you will be finding there will be now this nuclear membrane. Exactly I'll be telling you when it happens, don't worry, but this will be happening, and in the end cytokinesis will be happening. Cytokinesis means this will be breaking up over here. Now in animal cell there is no cell wall, so that's why there is cleavage furrow method which is being utilized by the cells. But over here plate formation method is utilized in the plant. Plate get forms over here, and then these two will be dividing. And how this will be dividing over here? This will be becoming one cell, this will be becoming one cell. This is nothing but mitosis. From one cell you have got this. Alright, now let me show you in this particular diagram also you understand. Interphase doubles. Prophase it got smoothened out. Metaphase kinetochore is getting attached. Anaphase they will be getting separated over here. Teleophase nuclear membrane, separate nuclear membrane will be happening over there. इसमें ढंग से समझ नहीं आ रहा आपको, इसमें overall दिखा रखा है. You don't have to draw it like this. Let me show you on animation. This you read out first, guys. Interphase, prophase, like this only you have to read. What is happening over here? Just pause the video and try to read. Right. Nuclear envelope starts disintegrating. So nuclear envelope starts disintegrating in the prophase itself. Right. Then a metaphase you will be finding over here. Metaphase, what it will be doing? You will be finding these. What is these? Fibers will be coming over here. Right. And these are made up of microtubules. And spindle fibers will be attaching to the centrosomes over here. Right. Kinetochores. And then they will be snatching away over here like this you will be finding. Okay. Then anaphase, what is going to happen? That over here the chromosomes then pull towards pole by the fiber attached to kinetochores. The centromere of each chromosome leads to an edge while the arms tilt. So this is snatching is happening in the anaphase. and in the telophase the membrane will be coming over here. Okay, this is fapring over here, nucleus, everything will be doubled over here, and then this particular part will be separated by cytokinosis. Okay, so see this over here, guys. This is the interface which you are going to see. Interface is containing G1, S, and G2. Similar way you will be showing it in the exam also, these three phases, and one is the mitotic phase. Cell division, this is cell cycle. Cell division method is the thing is the mitosis, where cell division will be happening, and the complete this is known as cell cycle. Okay. Now G1 phase, what happens over here? Things will start getting duplicating except the DNA. DNA will happen duplicating in the S part, synthetic, right? The cell is growing larger. Why? So that it can accommodate double cells. Okay, nerve cells will be getting doubled over here, small muscle cells will be getting doubled over here, right? See this, S phase will be coming and the chromosomes will be getting doubled over here, guys. Okay, just see over here. Let me just do it a bit fast. Okay, over here these chromosomes are doubled over here, not chromosome but the DNA is doubled, right? In the G2 phase, again you will be finding the left out things will be checked over here so that it can enter into the division phase. Division is mitotic phase. So you will be finding this thing will be happening over here, and then mitosis, the M phase will begin. See this M phase now over here. Okay? M phase you will be finding mitotic phase, M phase, mitosis will be having, एंड देन साइटोकाइनेसिस ऑल्सो माइटोटिक फेज में ही आता है, तो इसको माइटोटिक फेज भी बुलाते हैं. इसके अंदर दो चीजें, माइटोसिस, साइटोकाइनेसिस. माइटोसिस इज पीएम एट प्रोफेज, मेटाफेज, एनाफेज एंड टीलियोफेज. What is happening now? See clearly. This chromatin is getting, the double DNA is getting a bit condensed. Now it will be clearly visible, the double copies like this. You try to see. This is happening in prophase. Right, chromosomes like this you will be finding. Centromere in this I have already shown you. Sister chromatids I have already shown you this. Okay, now this will be clearly visible in this form, and from this particular micro, this spindle fibers are coming from the, you are finding this is happening from this particular centrosomes. Centrosomes will be going to the two corners. This nuclear membrane will be disappearing. It is happening in the prophase only. You try to remember. Right, in the metaphase what is happening now, that these are attached, spindle fibers are attached to the kinetochore and they will be assembling at the equator. That's why we are saying the best way to see chromosomes it is at the metaphase. Since starting I am telling you, metaphase you are understanding. Anaphase, now it will be stretching. Now they will be taking the copied DNAs at one end over here, and it will be a mix match, guys. Okay, it will be a mix match over here which will be happening. Exact same copies will be taken over here. Now these are taken over here. Anaphase happens. Now in the telophase you will be finding. Telophase you will be finding that these will be coming together now. Right. Centrioles are at the poles over here. Telophase you are going to find that the nuclear membrane will be coming. This what was that? Nucleolus will be coming over here, and the enzymes will be coming, and it will be forming a separate cell over here. Right. Now over here you will be finding the cytokinesis. In animal cell you will be finding there is a cleavage furrow. But animal cell, why? Because a membrane is there, no plants, no cell wall. But in plant cell you will be finding this plate formation will be happening. So they will be showing you the plate formation. Just try to see the plate formation. Cell plate we call it. This is a cell plate which will be forming, which helps. कहाँ से बनती है? Golgi bodies, endoplasmic reticulum functions over there, right? Send the functions. Okay, then two daughter nuclei will be formed. So I hope you have understood this in a better way. Now for the examination you have to write it in this way only, guys. You will be getting only 2 pages to write. Okay, and the question which has come in the examination in 2025, you will be finding that it was too broad. So you have to condense it in 2 pages because it was a 10 marker. Just see the question. They have asked you about the cell cycle and explain that cell cycle over there. Now comes the part of meiosis. Meiosis is also known as reduction division, because over here from 2n I need n haploid. 4 haploid daughter cells are formed from one diploid parent. This we have to understand. Now everything will be same over here too, guys. Okay, everything will be same. The thing which will be changing over here, that now the mitosis over here will be happening at 2 stages. Oh sorry, meiosis will be happening 2 times over here, right? So understand over here clearly that meiosis will be having 2 cycles basically in order to have 4 daughter nuclei. So one will be known as M1, meiosis 1 stage. Second will be known as MEM2, meiosis 2 stage. In M1 there will be same procedure PMAT, but you have to add 1 in front of them because P1, A1, M1 and P1 things will be happening over here. Similar over here PMAT, but 2 second cycle, like this you have to mention over here, right? The change where it is happening meiosis, the major change you will be finding over here in P1. And P1 you know it as prophase 1 of meiosis 1. In this prophase 1 also you will be finding several stages like P, Z, L, or you will be finding this D and D over here. P is for पैकेटीन, याद करना पड़ेगा, ज़ाइगोटीन, लेप्टोटीन, ये याद हो भी जाएगा गाइस एक दो बार करके ना, डिप्लोटीन एंड डाइकाइनेसिस, पैकेटीन, ज़ाइगोटीन, लेप्टोटीन, डिप्लोटीन, और इसका थोड़ा प्रोनाउंसिएशन अलग है तो डाइकाइनेसिस. Now understand what is happening. Packetine is same. Over here you will be finding that the cell is getting condensed and the chromatin fiber is getting doubled over here. ठीक है, यहाँ double पड़ा है यहाँ पर सब कुछ. ठीक है. In zygote what is going to happen? It is going to this all is happening under prophase only. ठीक है. Just wait. ना. What happened to this? Alright. This you will be finding like this. ना. So over here what will be happening that this particular nuclear and in this nuclear these will be getting divided like this. Suppose I am taking just 2 example, hai na? So it has got doubled also and it will be appearing like this. But in zygote stage what they will be forming, they will be doing the homologous pair. They will be forming like tetrads like this. They will be joining each other. For example, this is over here, so white will be joining towards it. Same fashion they will be joining like this. So they will be forming tetrad shape like this you will be finding, hai na? Now because of this certain thing will be happening in the leptotene stage. What will be happening in the leptotene stage? You will be finding that crossing over will be happening, and that is why we are different from our parents because of this stage only, prophase, leptotene, zygotene, because over here our chromosomes number, DNA is getting changed, crossing over is happening. Crossing over, what is that? In detail we will be understanding in separate lecture. But right now you just must be understanding that because of this, what is going to happen? That in the white DNA, some part of this particular yellow DNA must have come. And in the yellow one, some part of white must have come, crossing over. And then synapses will be happening. Synapses means they will be going apart from each other in the diplotene stage. Okay, like this they will be getting separated over here in the diplotene stage, and then in the dikinesis they will be coming at the plate formation like this. I'll be showing you like this. Okay, let me show you if I have diagram, otherwise I'll be showing you in animation. So see this particular part over here, prophase divided into leptotene, zygotene, packetine, diplotene and dikinesis. देखो यहाँ पर I have got confused, that's why I was confused over here. There is leptotene first, L should become first, leptotene. ठीक है, and this crossing over is happening in packetine. I'm really sorry, packetine. Really sorry I got confused. यहाँ पर आप इसको सही कर लीजिए. Leptotene, zygotene, packetine. ठीक है, now you see. Leptotene chromosome become thin thread-like. Zygotene homologous chromosome. This you have to remember. It will be forming synaptonormal complex tetrad structure. Packetine crossing over occurs at chiasmata. Diplotene homologous chromosome starts separating, remained attached at chiasmata. That I'll be showing you what is chiasmata inside. Like this you will be seeing that middle one they will be connected, but they will be detached from the sister chromatids part. Dikinesis, chromosome fully condensed, nuclear envelope breaks and spindle forms over here, and like mitosis you will be finding the same thing will be happening. Metaphase, you will be finding that they will be aligning at the equatorial place, and spindle fiber attached to centromeres of homologous chromosomes will be detaching. Anaphase, again same thing will be happening. These haploids will be setting towards at each pole over here, and these will be in the telophase, will be converted into two haploid daughter cells with nucleolus appears and nuclear envelope appear over here. Yeah, this is the first meiosis. In the second meiosis, the duplication will happen only. It will similar to mitosis, because over here copy has to be made of two n particles. So n and n will be made, and like this 4 daughter nuclei will be having. So over here prophase 2 will not be having leptotene, zygotine, pachytene, dd. It will not be happening, right? Over here simple, what has happened in mitosis, simple thing will be happening again over here. So let me show you once animation, you will be understanding it in a better way. See over here guys, reduction division. These are containing your haploids, and this has to multiply. How they will be multiplying over here, or how they will be creating at the first place, you will be understanding. Meiosis 1 prophase, PM at, you don't have to remember it again, already you have remembered it in mitosis. The only thing which you have to remember over here is LPZ LZP DD. Remember it again and again. So in prophase, you will be finding this diploid chromatin is there, and it will slowly getting condensed, linear. It will be easily shown to you, right? There you go. Now it will be converting into this. Sister chromatids, I have already told you. Now under the leptotene phase, not leptotene, but this is the phase of Z phase, zygotene. They will be forming synapses. Tetrad will be formed. They are not mentioning the inner particular phases, but I am telling you that also. These alleles like this, they will be joined over here. I hope you are seeing this. Then a crossing over will be happening, and this happens in the pachytine phase, this crossing over. And because of this, chromosomal aberrations happen, which we have to understand in the next lecture. Okay, like this, different recombinations are made. That's why the progeny of a particular parent cell is not reflected in the daughter nuclei, right? That's why they are separate. That's why we are different from our parent generation. That's why plants are showing different characteristics. Okay, so over here this will be formed in the prophase. Only in the DD they will be getting separated too. Now in metaphase, what you will be finding, that they will be spindle fibers will be getting attached, and they will be coming at the ends. First they will be coming at the center, and then they will be coming at the ends over here. And telophase, they will be forming the whole over here. Plate formation will be happening, and separate nuclei, daughter nuclei will be formed over here. Okay, so this will be happening. Cytokinesis will be happening. Again meiosis 2 will be happening, and over here one more time they will be going under. Over here no DNA replication will be happening. Simply just they have to divide over here, right? So metaphase, they will be separating each other, and then NN. That means haploid we have got 4 daughter haploid nuclei we have got over here, right? Okay, so like this, you just have to mention this in one word. Not elaborate detail is needed of these things. If you will be writing it, it will be showing the depth of your knowledge. Otherwise, in a lot of books, this is not shown even, right? So this way we have covered this mitosis and meiosis in this particular session, guys. Mitosis and meiosis is completed. Later on, I'll be telling you about that why now chromosome aberrations are happening. That you will now be understanding in the next class. We will be picking up. Just try to absorb this information right now. Okay, just check out the PPT now. This cell cycle has been completed. This synthesis completely completed. More chromosome structure completed. In the next class, this topic and this topic we will be getting over with. Okay, in the next lecture, 5th lecture, we will be getting over with laws of heredity, right? So this is all for this particular class, guys. I'll be meeting you in the next class. Thank you so much. Till then, revise and come for the next class.